So this is a micrograph of the colon, and as you can see from the slide, there is the commensal microbiota over here, and there is healthy colonic tissue. And the microbiota is separated from the healthy colonic tissue by a thin mucus layer. Despite the potential antigenic load of the commensal bacteria in the gut lumen, the bacteria and the host live together as two peaceable kingdoms. And for the longest time, it was thought that the gut microbiome didn't do much. They were just there for the ride, until a few years ago, experiments were undertaken where germ-free mice were colonized with a commensal microbiota, and those mice were found to have a transcriptional response within the colonic tissue, thus realizing that there's a dynamic interaction between the commensal bacteria and the host tissue. And this was the springboard to the establishment of the Human Microbiome Project, where they characterized the composition and diversity of the microbial communities on mucosal surfaces all around the human body, including nasal passages, oral cavities, skin, the urogenital tract, and the GI tract. And this characterization made us realize that we are one superorganism, the human and the microbiome. It's a realization that we're not just our cells and our genes, but we're intimately associated with our microbiome. And any exposed surface within the human body is colonized by bacteria. And in this slide here, we have a description of what kind of bacteria that are there. So we are a superorganism that's intimately associated with the bacteria in and around our bodies. And this also was a realization that a couple of years ago there was a paper postulating a new tree of life, and we and mice and other eukaryotes are placed down here in the tree of life, whereas your microbiome harbors a vast diversity of different viruses, of archaeobacteria, proteobacteria, and all different kinds of bacteria. So we truly are a super organism intimately associated with our bacteria. So another discovery of the Human Microbiome Project was that microorganisms play a vital role in human health. And an alteration in microbiome diversity has been linked to many diseases. And this is exemplified in the following slide, where dysbiosis of the gut microbiome has been implicated in many diseases. In such experiments, a group of healthy controls would be compared to patients. And in many of these diseases listed down here, there was a change in the microbiome diversity in these diseases. However, what is unknown is that whether the microbiome causes the disease or the change in the microbiome is as a result of the disease, i.e. correlation does not equal causation. So correlation in a microbiome diversity doesn't equal causation. So this realization has given the springboard to what's recognized as a second wave of microbiome studies, the transition from a correlation in your microbiome diversity to a disease to establishing causation, and specifically elucidating the molecular mechanisms by which the microbiota influences host physiology. And to undertake such studies, we require a gnotobiotic resources. So, and that's why we set up the Emory Gnotobiotic Animal Core. And the Gnotobiotic Core is located in the HRBC in the bottom floor of the HSRB, building. And we have a suite of germ-free isolators on the bottom floor of the building that look like this. Each one of these isolators can house germ-free mice. And if you look inside one of these isolators, it houses cages. Inside each isolator, there are 12 cages. And when we put them in, we had to put the cages in. We had to sterilize the isolator to kill any bacteria that's inside there. We then had to prepare water and food for the mice, which we prepare in packages. We have a precise autoclave regime that we use to autoclave the water, the food, the bedding, the paper towels, and everything that you need for murine husbandry inside an isolator. We then pass these packages into the isolator. There are these side ports on the side of each isolator. We put the packages inside the isolator. We spray Clydox inside the side port, wait for a few hours to kill all the bacteria on the outside of the package, and then bring the package through the inner port door here. And that's how we can give more food, more water, and more bedding, etc., to keep the germ-free mice alive inside the isolator. And when these packages come inside the isolator, they look like this, and they're sterile on the inside and the outside of the package, of course. And then we can bring the mice in, and inside one of the isolators, we can maintain a colony of germ-free black 6 mice. Okay, so that is how we can keep a colony of germ-free mice going, and we've done that for a few years here at Emory. But people ask, so we have black 6, but people want to also have a germ-free version of their own specific knockout mouse, for example. So the way to do that, if you have your knockout mouse, for example, an IL-10 knockout mouse, you would send a breeding pair of those knockout mice to Taconic Biosciences. They then do IVF to implant your knockout mouse into a Swiss Webster mother. We then ship that Swiss Webster mother to Emory and then establish a colony of your knockout mice germ-free inside our isolators. And this all costs about $7,500, including shipping. So the mice are transferred from Taconic to us inside these germ-free shippers, these sterile shippers, and these would be the germ-free Swiss Webster mice. We have to hook up this shipper to our isolators, and we unfurl the little pocket over here and hook it to our isolator. There is a little port on the side of the little tunnel where we spray in the Clydox. We then put a little bung there on the side of the tunnel and wait about a few hours for this tunnel to sterilize. And then we can break the seal over here and then pass the mice inside our isolator. So you will have Taconic ship 7-day pregnant mice to us. We bring in the Swiss Webster mice inside our isolator and wait for them to deliver. And most knockouts, of course, are on a black 6 background. So what we'll get is a Swiss Webster mother that gives birth to black 6 pups. So you'll know that you've re-derived your genotype because they're black 6 pups. And then hoping that you have males and females from the colony that's being delivered by the black 6 mother and by the Swiss Webster mother, and then you can establish your colony based on these from the knockout mice that was delivered. Okay, so that is making germ-free mice. But like I said, a lot of the utility that people are asking these days is defined microbiome or gnotobiotic mice. Remember a little bit on terminology. Germ-free means germ-free, no bacteria at all, whereas gnotobiotic means to know the bacteria inside the mouse. So you can put a defined microbiome inside the mouse and call it a gnotobiotic mouse. And for these research, we have acquired what's called a Techniplast isocage bio-exclusion system. So this is germ-free science, but on cage level. We can keep gnotobiotic mice inside these cages. So the utility of this, if you want to do a defined microbiome study, is the space comparison. If you wanted to do a precise microbiome study or a defined microbiome study inside an isolator, you would have to reserve a whole isolator with 12 cages inside to do the experiment. Whereas if you want to do the same experiment, we can use the ISO cages and just use one cage to do the experiment. And remember, these cages are hermetically sealed cages. Nothing goes in or out. And I brought one of these cages along to demonstrate what they look like. And I had to switch over to the video screen. I'm going to get the cage. So what happened, the cage looks like this, and it's opened like this. And you work with the mice, and then you reapply the cage, and then close the side seals, and then close these down, and then you put it back on the rack, and on the rack shown there. And when you push the rack in, it pushes in these little ports, such that air comes in through this port, goes through this sterile filter. There's also a couple of sterile filters in that gray part of that rack, and we've managed to keep mice germ-free inside these cages for a few weeks. So this is a major advancement in our technology, in gnotobiotic technology over the last few years. So I'm returning to the screen now for those watching online. So okay, so the workflow that we've established is we generate germ-free mice in these isolators here. We will then transfer the germ-free mice into these ISO cages, these hermetically sealed cages, for you to do your specific experiments, either colonize with a specific bacteria or a certain treatment or anything like that, and then do a gnotobiotic study in this defined microbiome environment for maybe 6 to 8 weeks or longer. We've actually gone 3 or 4 months and successfully kept mice inside these isolators. And like I said, some more images of the system. There's 36 cages, so we could theoretically do about 30 or so gnotobiotic experiments at the same time, whereas in the old days you would need 36 isolators to do have a similar research output. We handle them very carefully. We take the cages off the racks. We then open them inside our hood and basically treat them like cell culture, so we can keep them germ-free. And we rigorously test for azenic environment inside those cages just to make sure that they're germ-free. Okay, so like I said, demonstrating the whole science is going from a correlation of a microbiome diversity to demonstrating causality of a certain microorganism. For example, if you've done the microbiome analysis of disease patients to healthy controls. You might have found an organism that's enriched in your disease patient, and then you want to test whether that organism is responsible for the disease. And we can do these experiments by monocolonizing mice just with a single organism, monocolonizing a germ-free black 6 so that it just has that organism, and then you can infer causality of that single organism to the certain disease state or whatnot that you're looking at. So that's monoassociation with the one bacteria, and these would be housed inside those ISA cages. So if you have a wild type bacteria and maybe a null bacteria for a virulence factor, for example, you could do those experiments in a short amount of time as well. But the leading edge of the field at the moment to demonstrate causality is something called human microbiome-associated studies. So if a certain microbiome diversity has been associated with a disease state in humans, you can take the microbiome, collect the microbiome from those diseased humans, and then colonize a germ-free mice with that disease state, with that microbiome from the diseased patients, and then see if the disease phenotype is transferable within the microbiome. If somebody's claiming that the microbiome causes this disease, well, this would be one way of proving it. And again, we've done these sort of studies inside our ISA cages where we've inoculated germ-free mice with human fecal samples and kept them for a few weeks. And the microbiome diversity, with the caveats, of course, there are differences between the human GI tract and the murine GI tract, but we keep the diversity of the microbiome, especially because the mice are housed within these ISA cages. No other microbiome goes in because they're in a bioexclusion state and nothing can go out. And of course, mice are coprophagic, so they'll eat their own excreted pellets. So you maintain that microbiome that you've inoculated from that human patient inside these cages. So we have the capacity to do that as well. Okay, so those are some of the resources that, or some of the facilities that we have available. I will end by giving a list of the resources that we have for you, but I want to show a few slides now about microbiome diversity and how it affects experimental science. So, microbiota and reproducibility of mouse models, and why should you care? So there is the NIH, of course, have given directives recently for rigor and reproducibility, because scientists from various universities are getting disparate results from similar knockout mice, for example. So, and this was a big problem for a long time, and until it was realized that the diversity of the microbiome inside those mice was a contributing factor to variability between mice. So if you have a model organism, that you have a mouse, for example, and there's variability in experimental results, well, you want to make sure that you have model performance, that the therapeutic response that you're observing is faithful and reliable, and that your model has translational relevance. I'm going to show you a few examples from the literature, how people have found reproducibility or issues in reproducibility from facility to facility. This slide demonstrates the different factors that can contribute to variabilities from vivariums, from different vivaria. The microbiome diversity in the vivaria here is different to what it is in Vanderbilt or Duke or Harvard or whatnot. But really, not only that, we can have differences in the microbiome diversity from different buildings here at Emory. What's in the Whitehead to the Woodruff to the VA is different. So you want to control, you want to sort of just make sure that you account for any differences in microbiome diversity. And of course, these lists along the side here can contribute to variability. This was a study that was carried out a couple of years ago, showing how variable the microbiome can be. These mice here were purchased from Jackson, and these mice here were purchased from Rheinallt. They looked at the microbiome of 3 different mice strains purchased from the 2 different companies, 3 different strains from 2 different companies. And what they found was that the Jackson microbiome was similar in the 3 strains, and that the Rheinallt microbiome was similar in the 3 strains. So it looks like the microbiome is similar in mice, irrespective of the mouse strain. So that was the first thing that to take into account. Secondly, this experiment did a DSS colitis model in mice that they purchased from these different facilities over here, listed here. They looked at the disease severity in response to DSS, which induces chemical-induced colitis in mice, and saw that the mice from the different vivaria responded differently to the insult. So this is proof that there is variability in results from mice purchased from different vendors. A famous story of, regarding this, of course, is the induction of Th17 and IL-17 by bacteria called segmentus filamentous bacteria. And this was found about 10 years ago. Now, the story goes, the results was they purchased mice from Taconic and purchased mice from Jackson. They found that Jackson harbored this specific type of bacteria called a segmented filamentous bacteria. If you co-house the Jackson and Taconic mice together, these segmented filamentous bacteria will transfer into the Jackson mice. And this was important, of course, because these segmented filamentous bacteria, these SFBs, are potent activators of Th17 cell maturation and IL-17 production. So you got a bunch of mice that you bought from Taconic, they have high IL-17, and the ones from Jackson that didn't have the SFB, and they have low IL-17. And that, of course, would confirm experimental reproducibility. And what this is showing is that if you take the germ-free mice and colonize with this type of bacteria, then you can induce the IL-17 response. So the presence of certain microorganisms within the microbiota can influence host physiology and immunology. So what can you do about this is that, for example, within Taconic, you can order mice by location and health standard. There's a lot of information on the Taconic website, for example, about the different state that you want to buy your mice. And so it is, there is people, I was chatting to the guy, Phil Dubé from Taconic, and he said people think that all Taconic mice are colonized with this SFB. Well, apparently not. Taconic has about 40 facilities throughout the country, and only half of them have this SFB, microorganism that induce the IL-17. So when you buy in your mice, make a note of which vivarium from the company that those mice came from as well. We were recently asked by Cell Press to state in our materials and methods which Jackson vivarium we purchased our mice from, for example. So keep a note of where you get your mice from and which vivarium. So strategies for accounting for reciprocal host microbiome interactions in experimental science. So what can you do about all this? So minimize microbiome variability over time between groups. Strategies such as co-housing and using littermate controls and wild types that have the same microbiome in your approaches. What people have started to do now is fecal banking. When you sack your mice, you keep a fecal pellet from the mouse, keep it at -80, and know that at this time, during this experiment, that this was the microbiome that gave this phenotype. If you have a catastrophic event and you lose your colony, then you could rederive your mice and then reintroduce the microbiome that you kept from a few years ago, and then you can carry on with your research. So that's fecal banking. And then, as I alluded to, validate microbiomes by production location for model performance, that you have variabilities from different vivaria. And as I said, that what we can do in our facilities, deliver colonization of microbiomes to create custom microbiomes tailored for specific applications. We can raise germ-free black 6 mice, you can give them a specific microbiome, and you can house them within the ISA cages for specific microbiome approaches that you would want to do. So another few examples from the from literature. The microbiome within vivaria is very different to microbiome in the wild. So there was this paper in Cell a couple of years ago, which I like to show. These are the microbiome diversities from mice that were collected from barns in and around the Maryland, in and around the NIH. So they went out, collected the microbiome from mice that were living wild within barns in 3 locations in Maryland. They compared that microbiome diversity to the microbiome diversity in Jackson, in Taconic, Jackson, and Charles River. And as you can appreciate here, the microbiome of mice inside Bavaria is considerably different. What they did, like we could do over here at Emory, is they took the microbiome from these mice and colonized germ-free mice with these microbiome. And what they showed, that the wild microbiome protects against influenza A and a DSS AOM model of hyperplasia. So over here, they gave the wild type microbiome in green to germ-free mice and gave them influenza A, and they survived much better compared to the mice with the microbiome inside our labs. Similarly, the wild type microbiome, when given treatment that induces hyperplasia in the colon, they have much less hyperplasia. So take that into account as well, is that the microbiome that you have inside the inside our facility is pretty clean, and does it really represent the microbiome in people? And with all the heterogeneity within the human population, where we have different diets, different lifestyles, different habits, and antibiotic use, for example. Last example was that I like to share with you, and perhaps give you ideas to undertake experiments, is that this paper from Science last year that showed that distinct commensal microbial communities influence anti-PD-1 responses in patients. So anti-PD-1 is an anti-cancer drug. We give anti-PD-1, it limits tumor burden. This number of patients were given anti-PD-1 treatment. This number, 26, were characterized as non-responders to treatment, whereas this number, 16, were identified as responders to the treatment. Okay, so 26 patients responded to anti-PD-1, anti-cancer treatment, whereas 16 patients responded to it. They did the microbiome diversity. They analyzed the microbiome diversity, compared the non-responders to the responders, and found differences. And when you do a plot, the responders clustered together over here, whereas the non-responders clustered together over here. So inferring that the microbiome diversity within these patients had an influence on this anti-cancer therapy, this anti-PD-1 therapy. What they did afterwards was they took the microbiome of these responders and non-responders and colonized germ-free mice with them and gave the same anti-cancer therapy, and found that the phenotype could be transferred to the mice. So these are these are the responders, sorry, the non-responders in blue, and this is tumor volume. So irrespective if you give PD-1 to these mice, they still get high tumor burden. The responders given anti-PD-1 responded to anti-PD-1 and lowered their tumor burden. They looked at the microbiome over here, and in the responders had these bacteria enriched in them, and also these bacteria activated effector CD8 positive T cells within these mice. And of course, as you know, those type of cells function in response to cancer and malignant cells. So this is a great example of how they took the human microbiome from patients and non-responders and did an experiment in mouse to show that the microbiome functions in the host response to this anti-cancer therapy. Okay, so that's the final example I want to share with you. And I just want to remind you of the resources that we can provide at the Emory Microbiome Facility. So we can provide germ-free black 6 mice to use. So we have a breeding colony, and we sell those for about $300 each. If you want to buy them from Taconic, they're more like $500 each, and you would have to pay for shipping as well. And shipping in one of those shippers, those yellow shippers, cost about $1,000. So all in all, you know, we're about half price of buying germ-free mice from Taconic. We also, I told you about the control of rigor and reproducibility in experimental groups. We have an isolator that has this altered shadeless flora microbiome. And what altered shadeless flora is a community of 8 bacteria, which has 2 lactobacilli, and what's written down here, so it has 8 specifically defined microbiome of just 8 bacteria. So one of the problems with working with germ-free mice, of course, is that they don't develop properly. Their immune system doesn't develop properly, and the gut tissue, for example, is more fragile, and the villi are long and slender. So if you want to compare conventional mice with microbiome to germ-free, there are a lot of caveats. So somewhere in between, you can do your experiments in mice with this altered Schaefer-spora, these ASF mice, just with these 8 bacteria colonized, and they have a normal immune response. So this would be exquisitely accounting for microbiome as a variable in your studies. So as I said, we can house and establish breeding colonies of mice of a certain genotype that you want to rederive. So like I said, you send your knockout mouse to Taconic, we get pregnant females back, and we establish a colony of those mice inside our isolators over here. We could do defined microbiome experiments within our bio-exclusion cages, and those cages are housed in an ABSL2 facility. So if you want to use certain pathogens within these ISO cages, we can do that. So we can do that. As I said, we can do mono-association experiments in the ISO cages, and we can do humanization experiments as well to show causality of the microbiome. And finally, if you don't want to buy full mice, we also offer germ-free tissue to sell as well. So if you wanted to compare conventional to germ-free mice and look at the transcriptional response in the colon, for example, we could sell you 5 colon segments, 1 centimeter segments of colon, and you can purify your RNA from there and do qPCR, for example, to compare germ-free mice to your conventional controls without having to buy a whole mouse. We could just sell you a bit of tissue. And our prices and our facilities, everything that I've said, is under this website here. And if you want any more information, you can email us at feedback@emory.edu. And my final slide. So I'm the scientific director of the facility. Caroline Addis and Amanda Metzger, they run the day-to-day running of the facility, and they're really the experts in handling the germ-free mice. And also we have help from Chris Raymond as well. So thank you for your attention. I'll take any questions. Right, Mary.
Can you do different diets in the isolators? Do
So
you get them from the vendor autoclave, or do you autoclave them yourself?
So the question was, can we use specialized diets in the cages and in the isolators? And the answer is yes. So the diets that we use to breed mice is a special diet that we can autoclave and stands up to autoclaving and preserves the nutritional composition. So we use that. But if you want a specialized diet, what you would do is get in contact with a company that provides your specialized diet. And what we suggest is irradiating the specialized diet, because a lot of the specialized diets do not stand up to autoclaving, and they disintegrate. But what we've done with researchers is have them formulate their diet, then the company will irradiate that diet to a certain, they know exactly how much grays or rads that are required to make that diet germ-free, and then they package them into little brick-like packages, and then we can either give them to the ISO cages or pass them inside our isolators, depending on the scale of your experiment. So the answer is yes, we can do specialized diets.
Do you test for sterility, or would I need to do that?
The answer is, so the question is to test for sterility in the specialized diet. Yes, routinely we sacrifice one bag that you would get and use that as an example, sort of a test bag for sterility, and test for azemia or germ-free in the diet from one brick, if you like, of food.
Similar question is, could you give a compound any other way? Is it possible at all to gavage or IP inject an animal and keep it germ-free?
The question is, can we IP an animal or gavage? And the answer is yes. So the mice inside, we've been successful in keeping mice inside the ISO cages, and then we've given them oral gavage or an IP inside the ISO cages. We have a specialized restraining equipment where we submerge in Clydox and in disinfectant, and then we hold them by the tail and can do tail vein and everything like that. So normally, if an investigator comes along and if you want a specific procedure, then we work with an investigator to optimize a workflow that makes sure that the microbiome, the germ-free status is preserved, or the microbiome diversity is preserved. So yes, we've done that.
So I actually had a question along those lines. So for example, if we're keeping the mice for 6 to 8 weeks and we have certain procedures we want to measure a lot every week, so we would work with you or your group to make sure, just to give you an example, like motility, we measure like fecal pellet output or something like that. How would we, would we work that in?
So the question was, a 6 to 8 week experiment inside the ISO cages, and if you want to collect samples at time points every 2 weeks, for example, or weekly samples. So we would handle the mice because we're experts at handling the mice and keeping them germ-free. And then we give, with your assistance, we hand the samples over to you. And we have done experiments where we've kept mice germ-free or gnotobiotic mice inside the ISO cages. We've taken weights at regular intervals, weekly, over 6 weeks. We've collected fecal samples at regular intervals over 6 weeks. And so the answer is yes, we can collect weekly samples over the 6 to 8 ISO cage experiment duration.
Can the mice be infected with the virus in your facility, or?
So, like I said, we have an ABSL2 room. So I have to repeat the question every time to the listeners online. Can the mice be infected with a virus inside the facility? So the ABSL2 room is within the SPF facility, the specific pathogen-free. So any organism that comes into the HSRB SPF facility, we reach out to the vet, Vanessa Lee, to okay any microorganism that comes along. It depends on which virus it is. If perhaps the virus will compromise the SPF facility, then you would have to take certain procedures to isolate. But if it makes, if there's no danger of compromising the specific pathogen-free status of the facility, then I'm sure that sort of experiment would be allowed.
Second one, perhaps is on your website, and I should check that. But if we send our mice to Taconic or a particular knockout, and then comes back in your germ-free, you charge us per mouse or per cage in your germ-free?
So
how much that would be in us?
So when the mice come back from Taconic, and you want to establish a colony, then you would rent an isolator. Okay, okay, you would rent an isolator at 19, correct, kind of like $19 a day for an isolator to, and we do all the breeding, give them food, all the husbandry and everything like that. So once the mice arrive here, you're knockout blind, germ-free, you rent an isolator to maintain your germ-free colony at a cost of $19 a day. Yes. And any mice that are made out of that, they're your mice. We don't charge extra for those, of course, because you've already paid for the isolator. You would be, I think, charged for maybe an hour or so of just putting in the food or husbandry time or something like that, and that is about $130 just here and there over the month, just to husbandry charges. So, and we prefer for us to do all the husbandry because we've done it before and keep them germ-free for you, rather than having multiple investigators come into the facility and, you know, maybe be inexperienced in handling gnotobiotic mice.
And then if you want to perform any experiments with those mice, such as like giving them different diets or different, you know, infection, will they be done in that sort of, or do they going to go to the ISO cages?
So does specific experiments, can they be done inside isolator or ISO cages? So the answer is both. Okay. We would tailor the workflow towards your needs. If you have many mice, or if you have many different diets, or if you have many different fecal microbiome samples, for example, we would sit down with you and design the best workflow based on isolators and ISO cages. For example, if you want to preserve your colony inside the isolator, perhaps it's better not to introduce anything that would risk contamination inside the isolator. Take the mice out, your germ-free mice out of the isolator, and then do short-term experiments in the ISO cages, such that if you get contamination in the ISO cages, you don't have to rederive the whole colony. You can just go back to the isolator and get more mice within a couple of weeks rather than within several months. So that's the benefit of the isolator to ISO cage workflow. Shanti again.
Now, where are the conventional mice? Are we back in our own labs, or do you have?
Great. So which conventional mice would you compare to your germ-free? And that is entirely up to you. So,
so you have an ability to house the conventional mice somewhere close.
If you wanted, so we have the ability to house conventional mice. So again, we would design and tailor an experiment for you. So for example, if you have your germ-free colony established inside an isolator, what we would do, for example, is raise those mice to 3 weeks old to weaning age. We would take out littermates, and then we could colonize them with a microbiome that you want, that your conventional microbiome, and just house them inside the HSRB now, inside our facility or inside ISO cages, and then you would have littermate controls for your germ-free. That comes from the same cage, and then you've taken some of those mice out of the isolator and colonized them with a microbiome, and we can keep those in our facility or whatever microbiome you're interested in at that point. So you might want to colonize them with a microbiome from the facility that you worked with consistently throughout in the VA or in the Whitehead or whatnot. So we can sit down with you and design these experiments accordingly.
Right, I know you're with the mouse, but is there in your core facility, you can sequence the fecal samples, or is that run by you, or is it different?
Great question. So what about microbiome sequencing with Emory? So there is another core facility within Emory we have the genomics core run by Mike Zwick. So the genomics core can sequence the 16S microbiome, and then there is a pipeline. They've established a pipeline taking that 16S microbiome and sending it to the computational core that can do microbiome analysis. And that's not a trivial undertaking to do proper microbiome analysis using KIME and all these computational programs, bioinformatics programs. The facilities are here at Emory, so there's a workflow that you would collect the microbiome sample perhaps in the gnotobiotic facility, give them to the Emory Genomics Core, which do the sequencing, and then the Genomics Core will send the sequencing files to the computational core, and they will do the microbiome analysis, the 16S analysis. So yeah, all those resources are available at Emory now. One more question?
I just have a question about this, because we're doing a lot of work in cancer. So I wanted to know is if you wanted to put some kind of like model for tumors, is that possible? And also in terms of, because we do research in children too, for this kind of baby mice, how does that work in our core?
So the question was, can we facilitate tumor models in mice? So, you know, if the fundamental question that you're asking, like that last paper, does the microbiome influence the physiology or the disease state that you're looking at?